Welcome back, everyone. For those of you both joining us online and here in person in Sheldon Hall, thank you for all of your great engagement through the Q&A for those of you online and in the room asking questions. I think it's been really wonderful so far to see so much engagement and interest in these wonderful presentations. We will continue the morning, with a few more presentations. Our next presentation is from Sharon Patray, who's here with us in person from Johns Hopkins University. So I'll turn it over to Sharon. We look forward to your talk. Thank you. Here, you should be able to use it.
Good morning. I want to thank the organizer for the opportunity to present today, and everyone for coming out to listen to our presentations. So I'm Sharon Patray. I'm with the Synapse Lab, and I'll be presenting my work today on a study we've done where we are studying if the Golgi is essential for CSP trafficking, so the surface of the sporozoa for the sporozoite stage of the Plasmodium parasite. So as all of us in the room knows today, malaria remains a global health problem. It's caused by the Plasmodium parasite. In the Synapse Lab, we study the sporozoite stage of this parasite. It's the stage that develops in the midgut of the mosquito. It travels to the salivary, it travels and invades the salivary gland of the mosquito. When mosquitoes probe for a blood meal, it's injected into the skin. These parasites will then move in the skin, looking for a blood vessel where they will penetrate and be carried to the liver where they develop into the next stage. So sporozoites are known to be densely coated on their surface with the Circumsporozoite Protein, which I'm referring to as CSP. And as they move, CSP can be observed being shed as trails on glass slides. So armed with this knowledge, and due to the fact that we also observed that the Golgi in this stage of the parasite is relatively small, we hypothesized that CSP trafficking must be a Golgi independent process because it's shed in abundance as as the parasites move, and we know that the Golgi is very small. So to study our hypothesis, we took two approaches so far. The first is we did metabolic labeling experiments in the presence and absence of the Golgi inhibitor, Bifidin A. And then our second approach, we did super resolution microscopy to observe CSP colocalization with trafficking organelles like ER, Golgi, and we also looked at COP2 vesicles that carry cargo from the ER to the Golgi. So for our first approach, using Bifidin A, which inhibits the retrograde trafficking from Golgi to ER, this process indirectly inhibits trafficking from ER to Golgi. And in some cells, the Golgi disassembles and relocates to the ER, inhibiting trafficking going from Golgi to plasma membrane in the process. So using this compound, sporozoites were dissected from mosquitoes. They were incubated with S35 cysteine methionine, and in the presence and absence of Bifidin A for 3 hours. Our goal was to observe newly synthesized CSP. After the 3-hour incubation, we took advantage of the fact that these parasites shared CSP as they move. We spun our samples and we separated the sporozoite pellets from the supernatant containing anything that was secreted or shed into media. And then we took the sporozoite pellet, lysed them, and we immunoprecipitated CSP. And we also immunoprecipitated TRAP, which is a protein that is it's a microneme protein, and micronemes are thought to get their cargo from the Golgi. So we're using this protein as our positive control as a protein that we know go through the Golgi. We immunoprecipitated both our samples for CSP and TRAP. My first audio radiograph here is showing the CSP IP for both the minus beta samples and the plus beta samples. And we're looking at newly synthesized CSP in the sporozoite IP. And we're looking at newly synthesized CSP in the shed. And as you can see, both our plus and minus beta samples were shedding CSP just fine. And when we did the same experiment for TRAP, we see here that TRAP is synthesized in both our plus and minus beta samples, but TRAP is only shed in sporozoites that were not treated with bifidin A. Moving on to our second approach, looking at co-localization of CSP and TRAP as our positive control once again. We first started with the ER, looking at the ER. We used parasite lines in which the SEC61, which is a protein that coats the ER membrane. It's an ER membrane translocon. They were tagged with GFP, allowing us to observe the ER. These parasite lines were dissected, fixed, and stained for CSP and TRAP. The top panel shows the CSP, and the bottom panel shows TRAP. And as you can see here, we then analyzed the co-localization as the percent overlap between the CSP signal and the SEC61 signal. And that, based on that analysis, we saw that both CSP and TRAP co-localized with the ER at the same level. However, we did observe that the pattern of localization was different. So CSP here in the left two panels is showing more of a, it's localizing with the ER more at the periphery of the parasite compared to TRAP that is showing more of a perinuclear and vesicular pattern of localization. Next, we then looked at the CAP2 vesicles, and we looked at them by looking at parasites in which the SEC13 protein was tagged with a GFP tag, and the SEC13 coats the CAP2 vesicles. And when we stained for CSP and TRAP as well, we saw TRAP co-localizing to these vesicles to a significantly higher degree compared to CSP that showed little to no co-localization. And finally, we did this with grafts, parasites in which the Golgi protein grafts was tagged. And again, we saw TRAP co-localizing to a significantly higher degree compared to CSP. In conclusion, we feel significantly more confident saying that we think that CSP trafficking is a Golgi independent process. And for our future direction, we are thinking about doing experiments to show that will help us show how CSP is trafficked to the surface if it's not going through the traditional system of ER, going to plasma membrane. And for that, we want to do immuno-EM to help us look into this parasite at a more detailed level than super resolution has allowed us to. In conclusion, I would like to thank my entire lab, Dr. Patray, for the opportunity to do all of the study I've had the opportunity to do in her lab. Dr. Sashika Nitani, who worked with me on this project to do a lot of the beautiful images that you guys saw today. There's a name that I just realized I forgot to put up here, and that's Dr. Volker Herzler, who provided us all of the parasite lines that we used in the study. The rest of my lab members, the insectary and parasitology core, before we do any experiments, we need sporozoites, and sporozoites go to mosquitoes, and somebody had to help us do all that. And the microscope facility, as you can see, we have some beautiful images, and they help us facilitate all that. The NIH for funding my PhD so far, and MMI, and the Department of B&B, which is my parent department. Thank you.
Thank you so much, Sharon. We have a few minutes for questions, so please.
Hi, nice presentation. So I have a question. Do you think this difference in the localization of CSP versus TRAP, because TRAP is like more of a micronemal, right? So due to that, you see differences in their trafficking, or whether, TRAP is using Golgi mediated, whereas CSP is not?
I'm not sure I understand what.
The location of these two proteins are different.
Yes.
Sporozoite, right?
Yes.
So do you think because of that, CSP is not using the traditional pathway for trafficking versus TRAP is using?
Yeah, so while TRAP starts out in a microneme, they do eventually end up on the surface like CSP, and that's some of the reason why we chose it. It's also shared, as CSP is shared, although it looks like by different mechanism, and that's another reason why we chose it. As far as we didn't expect their interior localization to be different, as for a long time in the field, there were those who also thought that CSP went through the micronemes. But yeah, as far as the localization study, it's definitely been informative because they are localizing in different, it looks like in different regions as far as when they are trafficked to us anyway. Yeah.
Thank you.
Yeah, thanks.
Another question from Sam.
Yeah.
Rock tree biogenesis is also Golgi dependent. Did you observe any morphological defect in your rock trees by microscopy?
No, because in order to look at the rupture, we would need, at least by super resolution, we would need antibodies to it. And right now we don't have those antibodies, but maybe when we go to do immuno-EM, we will naturally be able to see the rupture even without antibody. And if there's any information there, yeah, we will definitely report it if it's something that come up. But no, we were not looking at the rupture to begin with.
Any other questions from those in the room or online? Great. Well, thank you so much.
Thank you.
Sharon.